Hi guys, how are you all? Welcome to our YouTube channel, which is Science Beyond Books, and I'm very happy that we are back with seventh episode of Science and Research Simplified. This is the show we do almost every Saturday, and the idea of this show is to bring researchers from all different field of science and talk about their research and give an exposure of research to school-going student and undergrad student. So if you are still not connected, try to join us, and then we'll start the show in a second. So our speaker is already here, and I'm gonna connect her soon. So let me have her on the screen, and then we can start the show.
Hello,
Hello, hi Harshul, how are you?
I'm good, thank you. And you? How are
And
you?
I'm very good. I'm very happy that you give us time and you want to share your research and give exposure of the field you are working in for a school going student and undergrad student. For them it's a great opportunity to listen to you and get inspired from your story.
Thank you. That's
So
also a opportunity to come here and talk about my work.
okay, perfect. So for the viewers, before we start the show, let me introduce you to our speaker. And the today's speaker is Dr. Harshul, and her background is she did her undergrad in biochemistry, right, from DU, Delhi University, and then she did her master's from Madurai Kamaraj University, and that's where I met her, and I know her from my master's because we are colleagues there. And after doing her master's in Madurai Kamaraj University in Tamil Nadu, she moved to Germany, right, to do her PhD. So it was Max Planck It's a very good, very known and very good university where she did her PhD. She had an outstanding PhD from there, and after doing her PhD, she moved to Switzerland, and she is doing her postdoctoral research from University of Geneva in Switzerland, and it has been almost 2 years, right? Since you started your postdoc?
Almost. Not yet. In July, it
Yes.
will be 2 years.
Yes. So she has been doing her postdoctoral research there in protein biochemistry, especially in the plant biotech section. Like, she's studying the protein associated in the plant. So that's what we have about Harshul. And one thing I want to congratulate her is she recently got her Nature publication. So for the viewers and for the student who don't know about the Nature paper, so when we publish any scientific work, we publish in a scientific journal, and there is a kind of ranking. It's not ranking like how we estimate that how good or bad a journal is. So we do it based on the impact factor. So the Nature is one of the well-known and well-respected journal in our field, and she had a recent publication. She published her PhD work in Nature, and very congratulations for you, because I know very few of our colleagues has any publication in Nature. So that's really outstanding, and I'm very delighted that you got a chance to publish your work in Nature.
Thank you very much. It's nature chemical biology, not nature. That's like really high.
Yeah,
So it's
so,
high.
I mean, that's your modesty, but it's still, having a paper in Nature is great thing. Yeah. So there is different kind of different, subcategory you can call it, or different sort of. So there is nature cell biology, and that used to be before nature NSMB, right? Nature structural molecular biology, right?
Yeah, there is another one. That's another one. This is chemical biology. That's the
NSMB.
nature.
Your voice is still not very, I think. Uh, Strong. But
Okay.
anyway, I think it should be fine. Viewers, if you have any difficulty to listen any of us, just let us know in the comment section. So, Harshul, before we start today's talk, I and even all my viewers would like to know that how you started, doing research. What motivated you to become researcher?
It's a funny story. When I was in school, I was always fascinated by the medicines that the doctors were giving. Why do we get one particular medicine for one disease, and why not another one would work? So, if you will see with my research, that's actually what I'm working on. It depends on the shapes of the enzymes which are catalyzing the reactions, and that's what I'm going to talk about today. So, this was one of the reasons that I took up biochemistry, because I was very interested to see why a particular molecule is a drug for one thing and not for another. Does it give you us? Yeah.
So that's interesting. Yep. So I mean, I think after after your presentation, what you're trying to explain is it will be more clear for viewers that what exactly you mean when you say
Yeah,
that different molecule have different treatment. Like, it looks, when you take tablet, they look similar, but they have different molecule inside, and
yeah,
different
absolutely.
molecule has different ways to treat different diseases. How it works, like how it happens. That's what you're going to explain in the today's presentation. So would you mind to open your presentation, and we'll try to keep it
Yeah,
interactive. So for the viewers, if you have any question, drop your question in the comment section. We'll try to take your question, and feel free to ask any question you have, and try to keep it interactive. If you want to come on screen and want to share the screen with us and ask question directly from Harshul, you can do that. Let me know in that comment section. I'll provide you a link. So you can click on the link and you can come on the live stream and you can ask directly question from the speaker. So you can always do that. Just let me know in the comment section. And for the viewers who are new for this channel, we do this show every Saturday. We bring researchers from all different field of science. So do subscribe to our channel and hit the bell icon so that you get notification for other talks. I think the presentation is ready here. So let me add the presentation to here. Okay. So Harshul, if you want to start, we can start
yeah,
because your
sure.
presentation is already on.
Okay. So the title was Structure and Function of Enzymes, but I'm going to be talking about proteinaceous enzymes. So in bracket, I'm writing proteins. Thank you very much for coming here. My name is Harshul Arora. The work that I'm going to talk about today is what I did during my PhD in the Max Planck Institute for Developmental Biology in Department of Protein Evolution. So to begin with, everything that is happening in your body, be it digestion, be it talking, be it seeing something, that all is happening because of a lot of chemical reactions. And for example, the chemical reactions you have, like it can make from A to B, right? There is a substrate that is A here, and the product is B. So if these reactions would have had to happen without the help of anything, they would be very slow. That's why we have catalysts which increase, decrease, or regulate the rate of the reaction. So most of the reactions that are happening in our body are happening at the speed because we have the catalysts called enzymes. Most of the enzymes, if not all, are proteins. So I'm going to talk about proteins today. And the reason why the enzyme works like this is because it has a certain shape. So, for example, if this is the shape of the enzyme, it can bind to its substrate because of its shape. And which molecule can bind to it depends on the shape of the molecule and the shape of the enzyme. So here you can see they bind like a lock and a key. And when then the product is formed, the product has a different shape. And that's why it can be released. So this is the reason why the shape of a protein is very, very important. And this is what is deciding which kind of medicine you are getting for which kind of disease, because almost everything that is happening in you, in bacteria, in plants, in fungus, any organism that you can imagine is happening because of different enzymes. And all of these enzymes have their own shapes. And some of the enzymes are not present in you, but in bacteria. And to kill those enzymes in bacteria, we give the compounds, the molecules which are binding to these enzymes and stopping that reaction, but it doesn't harm you because you don't have that enzyme. So in this way, the shape of the enzyme is very important. And how do we see this shape? Now, these enzymes, most of them, which are proteins, are made of building blocks called amino acids. The amino acids have two groups. The left side, the amino group that you can see, it behaves positively inside our body, and the carbonyl group behaves negatively in our body. And what happens is, because one is positive and the other one is negative, they can connect to each other. And then they form long chains. But, sorry, we went a bit ahead. But after this long chain, the chain doesn't stay like floppy long chain, it folds onto itself. And this folding is what is giving you the shape of the protein, which we just talked about. So the basis of this shape is based on the fact that everything in our body is in water, is in liquid. So they form hydrogen bonds. And the two main shapes are alpha helix, that's how it looks like, and a beta sheet. So these are just the models. We cannot see the original proteins. They are so small that you cannot see them by naked eye. So we have very special methods to see them. That's what we are going to talk about today. So the two main the shapes are alpha helix and beta sheets. And then they connect to each other in different ways and forms to give rise to higher shapes. So I'm showing you the shape of a very small molecule here, very small protein. It's called lysozyme. It's a protein which is it's an antibacterial protein. And as you can see, these alpha helices and beta sheets are connected together with some loops. And that's how more or less a protein looks like. And here I'm writing about a website called PyMol. If you go there, if we have time, we can look at it together. You can download the software, it's for free. And then you can download the files and you can look at these beautiful shapes yourself. You can change the color of them, you can play with them. We can do that if we still have some time today. Okay.
So Harshul, can I add something here?
Yeah.
So for the young student who don't know much about the protein structure, basically, so as Harshul very nicely explained that the protein is made up of it's a polypeptide, it's a chain of amino acid, right? The
Yeah.
sequence of amino acid decides that how it's gonna fold in 3D structure. And then the way it folds, like how many alpha sheet, how many beta sheet alpha helix are there in the structure, how they are organized, like two helix, then one beta, then one helix, like the way they are organized in 3D space, that decides the function of the protein.
Yeah, yeah,
If you
that decides
change
the
it a
shape,
little bit,
the
if you
function.
change
Yeah.
it to, like if you move one helix from another helix little far from each other, or just do any kind of change in this 3D structure, what you are seeing in the PPT, it's gonna change the function of the protein. And that's
Okay.
why many proteins become mutated, they become non-functional, they cause disease, because that protein is important for some role, they have some specific function. So if the protein's structure changes, their activity will change, and if their activity change, that's gonna affect you in many ways.
And
Right, Harshul?
yeah, right. And what can also happen is if you try to change the shape of the protein can also be dead. It can also kill the protein. It will not be functional anymore. That is
Yeah.
another scenario that can happen.
True, true. So even a
So
slight change in pH, slight change in the microenvironment in which the
yeah,
protein is, because proteins are present in the cell where you
yeah.
have hundred and thousands of different proteins. They live
Mhm.
together and they have a local microenvironment which has a specific pH, specific buffer condition, all those kind of stuff. Even if you take out the protein from those conditions, some of the protein they crash out, they
Yeah.
precipitate. Like one of the example you can understand with the milk. Milk has a specific protein on it. When you put a drop of lemon to it, it just precipitates the protein shape structure, that changes, and it
Yes.
just precipitates. That's
Yeah.
one of the example to
Yeah.
understand what's happening inside the protein, right?
Yeah.
Yeah, go ahead.
Okay. So since I was telling you these proteins are very small, you cannot see them with naked eye. So one of the techniques, there are many techniques, which time will not permit to discuss all of them with you today. So I'm going to start with X-ray crystallography. That's what I mainly did. So what we do is we try to make crystals out of protein. You have seen that when you take water and you keep adding salt to it, salt to it, salt to it, and then you leave it off for some time, the salt crystals might form. In the same way, I will come back to this again and show you in more detail how do we make crystals. But what we try to do is we take a protein out of the cell, try to keep it in an environment in which it's still stable. Most of the proteins are not happy, but if the protein is behaving properly, then you check if it's a correct protein, and then you try to make crystals out of it. Then what you do is these crystals have certain shapes, right? It could be cubical, it could be hexagonal, it could be rhomboid. And what we do is then we pass X-rays through these crystals. And when we are passing X-rays through these crystals, we get a pattern. That pattern is dependent on the shape of the crystal. And using that pattern, we try to understand what was the shape of the crystal. And then, because we know the sequence of amino acids that are present, sequence is not a problem, we can easily find that out, but we do not know the shape. So when we get sort of a shape from the diffraction pattern of the crystal. Then we try to fit the sequence of amino acids in that shape. We make a map called electron density map, and we try to fit the amino acids. And in that way, we can get the model that I just showed you. So there are many factors here. First of all, you have to get the protein, which is working fine, which doesn't, uh, which behaves properly in the environment that you're trying to work in. Then you have to have a crystal, which can take a long time. Then you have to have a crystal which gives you a good diffraction pattern that can again take a long time. And then you should be able to solve and get a map out of it. If you don't have a good diffraction pattern, the map might not be good. Or it could be that even if you get a good map, sometimes it's a very big protein and then you still cannot fit it. So there are a lot of how do you say problems here to get until the final protein model, and we will go through each one of them step by step now. Before I do that, I will tell you about the pathway I was looking at.
Can I ask a quick question before we jump into the next slide? So, why someone want to solve the structure of the protein? Is it just curiosity to know how the protein looks like, or what are the reasons scientists are looking for the structure of protein? Because it's a
So the
vast,
structure,
many scientists.
yeah, the structure helps you understand the function. As I was just showing you in the first slide, that the enzymes bind to the substrate because of their particular shape. This shape is defined by the structure of the protein. So you have binding pockets, which can only be made because of how the protein is folding. As we just discussed, that if we change that, the protein might not be active, the function will change. So the structure basically tells you the function. And that's why, to understand, to make new medicines, to make new vaccines, you have to first understand the structure of the protein. That's how you can change it, stop it from working, and you can play with it. But before that, you have to understand how it looks. Did I answer your question?
Yep. So just to add a few more sentences to it is very beautifully Harshul explained that how the structure is important to understand the function of the protein. One aspect she was talking about is to develop drug against it. So I just want to highlight a little bit here because my expertise is also the same field. I work on the drug discovery. So the protein I work is it's called RAS protein, and it's a very culprit protein, causes cancer. 30% of cancer happens because of this protein. Now, if we know the structure of the protein, and if we know that what part of the protein is the active part, like in our body, entire body cannot perform all the functions. We use our hand to grab something, we use our eyes to see something. So similarly, protein has different part and they have different function. So we know that this part of the protein is important for its activity, and that makes this protein active all the time. So if you design a small molecule, if you know the structure, when we say structure, we know the atomic level structure, we know all the atoms and their organization. It's not like just a helix or the ball. We know one molecule, each molecule, we know how they are organized, right, Harshul?
Yeah.
We know a complete map of entire molecule, so we can zoom in, we can see that what is the how the active pocket or active side of the protein is made, like what amino acids, what atoms are there, and based on that we can design a small compound, any compound which can go and bind there based on our logic that there should be some interactions. And if it binds there, it can block the activity of the protein. And if you block the activity of protein, basically you will not allow this protein to get hyperactivated, which cause cancer. So that's how you can
Yeah.
tackle the cancer. So that's one of the aspects. Yep.
So two things you can do. One is that you can make a compound which can bind more strongly than the original substrate. So basically, when it binds more strongly, there is a competition happening, and then the
True.
original substrate cannot bind, and
Hmm.
that's why you're stopping the reaction. The other thing you can do is how you make this compound like binding more strongly is because you change some of the you modify the compound itself, so somehow it is binding better to the pocket which is being
Hmm.
made.
Hmm.
So in that way you can make compounds which are binding better and hence stopping the original reaction that would happen. So for that you
Yep.
need to understand the structure,
Yep. So
and
the
you need to have
moral of the story is to know the structure is very important most of the drugs, are developed based on knowing the structure. So that's very important, and that's what Harshul is going to talk about, that how she solved the structure of some of the key protein during her PhD studies.
Okay. So I worked on chicken. Sorry, what were you saying?
No, I said that that's what you're going to talk about. That what protein you worked on, what structure you solved during your PhD. That is something you're going to talk about later.
Yeah.
Yeah.
So I worked on shikimate pathway. Shikimate pathway makes this corizomate, which is this purple compound here, and then this makes later a lot of vitamins, a lot of amino acids. So it's an important compound. I will be talking about only this pathway now because that's
Mhm.
what I worked on. Don't get scared with all these chemical names. I'm not going to talk about that. I just want to show you that this pathway has 7 steps. Okay, and the end product is corizomate. And why am I showing you this pathway is because normally, so the things written in bold here are the names of the enzymes which are catalyzing these reactions. Normally, what happens is that one enzyme does one reaction, but this pathway is a bit special. You have one enzyme in fungus which is doing 5 reactions. So it's one protein which is doing 5 different reactions.
In the same
That's why,
pathway.
yeah, in this of the same pathway. So out of 7 reactions, this one single protein was doing 5 reactions. This pathway is present in from bacteria until plants, but it's absent in humans. So we need to get these compounds from our food.
Mhm.
And that's why it can be used to treat some fungus and bacteria. So if we make drugs which can target this pathway, because it is present in humans, you can actually target the bacteria and the fungi which are doing this pathway, and they need it. Otherwise, they will not survive. Okay. So these 5 steps, step number 2 to 6, are happening with one protein molecule. This protein molecule, it's called arome complex. It was discovered in how do you call it? It was discovered in 1950s, but so far nobody was able to solve the structure of the protein because it's really big. As you can see, normally it's one protein, one reaction, but when it's doing 5 reactions, it means it has to be a big one. And the reason why we were interested in solving the structure was how are these? Is it possible that there is a channel between the enzyme, that the product made by the enzyme number 2 is like really going through a channel to the enzyme number 3? How are they arranged? Is it helping? Is it making the enzyme faster? Can it tell us that maybe other enzymes, even if they are far apart from each other, maybe they come together to make a shape like this while doing the reaction? So for that, what we did was we looked at the DNA sequence which is making this protein. So normally in a cell, you have a DNA which makes a protein. Okay, without going in a lot of details, DNA is the one which is deciding the sequence of the proteins. And this is the DNA sequence. And as you can see in the sequence, the step number 2 part is next to the step number 6 part. So with the sequence, we don't get a lot of information because this doesn't make sense that 2 and 6 are next to each other. Then it actually has to go after the reaction out from 2 and go and find that number 3 and then go find 4. So it doesn't really make sense. That's why we wanted to see if we can solve the structure of this protein and understand whether this huge machine is of any help or not. So for that,
So can I
first,
ask, Harshul, can I ask you one question?
yes,
So this protein, what I can see here is the numbering you have mentioned is it the amino acid number, or it should be amino acid number, right?
yeah,
One,
this is
two,
the,
eight.
yeah,
So it's like it's like pretty big protein, right? How many
yes.
amino acids you have?
Oh, how many? 1,500? 1,500, I think. 1,500 something, yeah.
Is it a one protein, or it's a complex of protein?
It's one protein. It's one protein. You can have it as, yeah, it
These are
was one of,
different domains of the proteins.
sorry?
And the color coding you have shown, these are different domains of the protein, which is
These are different
responsible
domains.
for
So in bacteria,
of the
for
reaction.
example, we have them as different proteins. Okay. In
Hmm.
bacteria, they exist as different proteins, but in fungus, this exists as one single protein.
Interesting. Okay.
Yeah.
Okay. Go ahead.
Okay. So
Hmm.
the only other protein which we knew was the one from plants, which has the only other bifunctional protein, one protein that can do two functions. It's also present in plant in this pathway. Step number 3 and 4 are making one protein and doing a reaction together. That makes more sense because you really know that it will definitely pass the product to this step. But other than that, we didn't know much about other multifunctional enzymes in these proteins, in this pathway, and no other structure was solved. This structure was known, and if you go to, we have a protein database. That's what I'm mentioning here. So PDB means protein database, and ID is the identification number. So we keep a record. So when somebody solves a structure of a protein, they deposit it in this database, because then other people can use it to solve other structures and use it later to make different medicines, make different vaccines. So every time somebody solves a structure, you share it in this database. And when you go to this database, you can look at it yourself. So what I did was I put a tag on I took the DNA sequence, I designed it such that I put a tag next to it. Why did I put a tag? Is because with this tag, I can fish the protein out. I will tell you how. So then I put this tagged DNA sequence in the bacteria, and we modify the bacteria such that the bacteria can act as a factory just to make our protein. If you want to talk about this, we can discuss this after the talk. How does this happen? So the
I think
bacteria,
that's what recombinant DNA technology is, right?
yes,
Okay.
yes. So as Ajit said about the recombinant DNA technology, we insert this DNA in a vector in the bacteria, and then we can induce the bacteria to overexpress our protein. Then what we do is we put it on a column, and that column can bind to this tag. Okay. And
Hmm.
then we are putting a lot of stuff because it will have other proteins that the bacteria needed to survive. The bacteria will make a lot of our protein, but it will also have, when we will split open the bacteria, there will be a lot of shit that we don't need, a lot of things that we don't need. So what we do is because of this column, when we are passing some solutions on this column, the tagged protein can bind to the column. And then what we do is we try to wash the column with things that bind to this column more strongly. And then in that way, we can get our protein out. So first, we bind the protein to the column. Then when everything else is gone in the washing, we pass the solution which binds more strongly to the column. Then we get the protein out. And we attach the columns to this machine that you can see on the left side. It's called an Acta purifier. It's a fancy pump, and you can regulate the flow of the liquids more sophisticatedly than what you would do with a normal pump. So the bottles contain different solutions which you pass depending on whether you want to bind the protein, whether you want to wash the column, or whether you want to elute your protein. You collect everything that is coming out of the column, and then you put it on a gel. And before we put it on a gel, we put really small amounts on the gel, not the whole thing. We take very little amounts. And then we kill the protein with a detergent so that the protein travels based on its size. And then we put a marker. You can see the sizes here. We put a marker, and then you can see if the size of the protein corresponds to the protein that you were looking for. In this case, this was a 170 kilodalton protein.
Hmm.
And you can see that in the elution fraction, most of what we are getting is our protein. Okay. Then what we do is we uh, take it, and if then there are other methods to check the integrity of the protein if it's fine, and then we try to crystallize the protein. So this protein is 170 kilodalton, and then it exists as a dimer, so it attaches to itself. So the total size of the protein is 340 kilodalton. So it was really a very big protein. So
So, just Harshul, can I just quickly summarize what you just explained in this entire slide? So, what Harshul is trying to say for very young kids, like most of the people understand what she might have understood that what she was explaining for very young kids. So basically, Harshul wanted to study this protein. She wanted to solve the structure of the protein. So the first step was to get the protein, pure protein in the hand. The protein is expressed in bacteria or in yeast, but she need a clean, clear protein in a
and a lot,
solution, in a tube, right?
yeah, and a lot of it.
And a
Yeah.
lot of protein, which so that was the sample she required to so that she can crystallize it. Like if you want to have a crystal of salt, you need salt. If your salt is mixed with, you know, seven different thing, the first thing what you want to do is you want to separate out the salt from all other thing, which is what we call purification. So when we force bacteria to make this protein, they have hundreds of different, you know, protein. So what she wanted to do is she wanted to extract that protein from all other bacterial protein, so that she has a very clean protein and in good yield. So that's what she did using this ECTA machine. And once she had that, she wanted to check the purity, that how pure her protein is. And that's what she did using the gel, because in the gel, if she sees just one band, one clear band, she don't see other proteins, which means it's clean. If she see five different bands, which means there are five different proteins. So using this gel, she was able to verify that she has a very clean protein. Now she was ready to crystallize it, right?
Yes.
Go ahead.
So for crystallization, if you can see this plate
Hmm.
with the blue background, that's the plate that we use for crystallization. It is a
Hmm.
96 well plate,
Hmm.
and we try, I don't know, 10, 20 plates like this with a lot of different precipitants, a lot of different salt concentrations, a lot of different additives to see which condition will help in the crystallization. The small, really small wells, you see, that's where we put our protein, and next to it we put this precipitant, high salt solution, and then because of vaporization, you might have some crystals coming. And we are talking in nanoliters here. That's how little volumes we are dealing with, so it's impossible to do it with hands. So we have robots for that who make the plates. So you put your protein solution and your precipitants in the you have one plate with a lot of precipitants. The robot takes the precipitants and distributes them in the plate, and then it pipettes your protein solution and distributes it in the plate. And then you wait for the moon to change, you pray to God until you can get a crystal. Because sometimes you don't get crystals. Most of the time you don't get crystals. And even when you get them, they are very bad crystals. They are salt crystals because we are using high salt concentrations, something the salt itself crystallizes rather than the protein. Because a salt is a smaller molecule, it's easier for salt to crystallize and it forms better crystals than the protein, which is such a big molecule. Not so big as you can see, but it's still much, much bigger than your normal salt. So it forms like the crystals, the quality of the crystals to begin with is not as good as it would be for a salt crystal. So you really have to wait very patiently. The crystallization for this protein took me almost 2 years. Okay, then
Excellent. So,
then this, yeah,
Harshul, can I add just one more thing? So,
yeah,
for kids who don't know about this whole process of crystallization, sometimes it's very challenging, and
yeah,
it might be looking very straightforward, as Harshul explained, but for many people, for many protein, it takes years and years to get crystallized. One of the example, if we talk about, is the way DNA was crystallized for the first time. And for the crystallization of DNA, the scientists, Watson and Crick, they were able and Franklin, Rosamund Franklin, they were able to crystallize DNA. It took many years for them to crystallize it. And once they were able to crystallize, that's the bottleneck. That's the most important part, is to crystallize the protein or any molecule you want to study. And once they crystallized, they were able to solve the structure, and that's why they got the Nobel Prize. And even
Rosalind,
make.
sorry, I have to interrupt you here because this is like, yeah, I have to interrupt you here. Rosalind Franklin was the only one who crystallized it. Watson and
Yeah, yeah.
Crick saw the structure from her data. Watson and
Yeah,
Crick
that
didn't
yeah, there
crystallize
is a lot of, yeah.
it.
So I did
Yeah,
not, yes, I did not wanted to get into that because
yeah,
many young people don't know about that. But yeah, that's true. So what happened is, she was the one. So she was the one who did the whole hard work to crystallize the protein, and then once it was crystallized, their seniors, with whom she was working, they got most of the credit for
yeah,
solving the structure and got the Nobel Prize. And there is a lot of backlash about it, that the
yeah,
way
yeah.
she was treated, and did not get the credit
No,
for it. But I,
yeah.
I did not wanted to get into that. Just get the
Sorry.
gist of the idea
Yeah,
that the crystallization is so important, and it's so, sometimes it's not very straightforward. Like I was giving you another example for the the viewers, that when some of the recent Nobel Prize was given for the crystallization of protein. The example is one of the Indian scientists, who is Venki Ramakrishnan, and she got Nobel Prize with Ada Yohanan, who is so these two wonderful scientists, they solved the structure of ribosome, which is the part of a, so it's a protein complex. It has
huge,
many protein,
huge.
50 55
Yeah,
proteins, they come together to form ribosomes. They solve the entire structure of ribosome, and they solve individual proteins in the ribosome, and that fetch them Nobel Prize. So it's not straightforward. It's a lot of hard work to get the crystal done, like to get the crystal happen. So that's why, as Harshul was explaining, that you have to try hundreds and hundreds of different conditions, because you never know which condition will give you crystal. And if you get the crystal, it's like baby, you got rewarded, like it's because I also did my PhD, and during my PhD I was trying to crystallize one protein. I'll tell you one story. And it took me like one and a half year to crystallize it. It was not crystallizing. So I decided to use another method, which is called NMR, to solve this structure. But just one it's very interesting to tell you that. So I almost gave up, that it's not going to give me crystal. I used to go and pray, "Oh, please give me crystal, man, it's not coming up." So I almost gave up. And then after 2 3 months, I just randomly went to check out of curiosity, like, let's see what's there. And fortunately there was crystal. Harshal, you might not know this. And there
yeah.
was crystal in one of the conditions. I was like, what? There is crystal. And then I shoot it in, and it diffracted to 2.3 angstrom.
Ooh.
Then we just did some more work on it, and finally we got 1.8 angstrom structure, and that gave me
Nice.
one paper during my PhD. So yeah, crystallization takes time. It looks straightforward, but if you get the crystal, because if you get the crystal for all the proteins, you can solve the structure of all the proteins, and you'll have a lot of information. But there is a lot of effort going on to crystallize and solve the structure of protein or protein complex, to understand the function of those proteins. So it must be a lot of hard work from Harshul. I'm pretty sure about it.
Okay. So once you get the crystal, as you can see in the plate, that it's so small, you have to look in the microscope to see if there is something inside. And then when you look at the top of the picture, there is like this loop, which is showing you the crystal inside. So that's how you have to take it out. And you cannot see now, the loop is shown from the microscope. So you can see how the crystal looks inside it, but you cannot even see this loop. So you really have to look in the microscope and then find the crystal, and you have to be fast so that it doesn't dry, because the we are talking nanoliters of volume. As soon as you open the plate, you seal the plate, of course. As soon as you open that particular condition, it can dry up. So you have to be really fast. You have to go there, and without destroying it, you have to get the crystal in the loop. And that has to be done properly, because if it is not in a particular orientation, you cannot get good data out of it. Now that you have this crystal, let's say that you got it, you have it in the loop, then you freeze it, because the crystal is just trapped in this bubble around in the loop, and it will start degrading. So you have to freeze this crystal as soon as possible. We freeze it in liquid nitrogen, so it's really cold. And then we go to the, because we need very, very strong X-rays. We cannot do it in our lab. We travel down to a facility which has which is called synchrotron. And you can see it in the second picture on the left here. It's a very big ring, which accelerates the electrons at a very high speed to uh, get the get the strength of the X-rays that we need to get the structure of the protein. Why do we need so strong X-rays? It's because there are a lot of hydrogens, and you can only see hydrogen when you're close to 1 angstrom. To get between 1 to 3 angstrom, after 3 angstrom, you cannot see anything. You need very strong X-rays. And the protein crystals, as I already told you, don't behave so well as salt crystals. So if it would be a salt crystal, you wouldn't need such a high intensity source. But when you're talking about protein crystals, they degrade very fast. When you're trying to shoot them, they degrade. And you cannot spend there for a lot of time to shoot them. And you want to collect, depending on the shape of the crystal, you decide how many times you want to rotate it to collect the data. And if you're trying to collect 720 degree, like rotating it twice, you have to be really fast. So you need a very strong source that can help you collect the data faster. In the old times, it was really hard. And that's why when people started working on it, it took much longer for them to solve the structure, because what they were collecting was not so good enough, and they had to sit there for hours to collect the data. But now it is getting faster, and at these sources, at the synchrotrons, you can collect your data within minutes now. So I don't have a picture of how the X-ray beam looks like from inside, because now nobody's able to go there because of Corona. You just ship your samples and the robot does it for you. But I have a picture of how a synchrotron looks like from inside. So if you go inside the ring, you can basically see all around you is the accelerator, which is accelerating the electrons, and you can walk around them. And then there are some places in which you can take the beam out, and they are called beam lines. And that's where you put your crystal and shoot it. And you shouldn't stay in the room because it can kill you. So there are like a lot of buttons that you have to press. Okay, there is nobody is in the room, and then we lock the room, and then we come outside and we start the rays. And then when you start the rays, as I already told you before, we collect the shape of the crystal, and then we try to fit the sequence of the protein in that. So for the arum complex, when we could finally solve the structure, we could see that it is actually existing as a dimer. And if you would go to PyMol, and you type this or arum, you will find the structure of this protein if you want to look at it yourself. So even though in the sequence of the protein, we could see that 2 is connected to 6, here we could see that this is the step 2. And if I show you like this is the step 2. It can pass the substrate to step 3 very easily, right? So even though in the sequence we didn't see that this would help, and these individual domains were not so close to each other, but in the shape, we can actually really see that this is helping in catalyzing the reaction. And then this can easily pass it here, which is the next step. This can easily pass it here, which is the next step. This can easily pass it here, and this can easily pass it here. So in this way, we could see that this is actually, it's not a channel, which we were thinking that it could be, because some proteins really have a beautiful channel inside them. It didn't have a channel. It was just bringing the different substrates in close proximity to each other, so the reaction probably could be faster. That's another story for some other day. We didn't find the reaction to be much faster in the end than other proteins. So we know that this protein is bringing the active sites closer, but it is not helping in increasing the speed of this reaction. So it could be just to isolate it from all the other different things which are happening in the cell, because there are thousands of pathways that are happening in the cell. Maybe to isolate that this protein was existing like this to bring the substrates closer. So with that, as we were talking about the paper, that's how the paper looks like. You can find it on the internet if you look for it. And yeah, so this was the paper that came out with this. And with this, I would like to thank everybody who helped me with the work. And if you still want, we can look at the thymol together. So I did this project with Dr. Marcus Hartmann. And the funding was from Professor Andre Lupas, and a lot of people helped me with this. And we were collecting the data at the Paul Scherrer Institute. And yeah, with that, I think I'm ready to take questions, or we can look at the structure in the PyMol, whatever you want to do.
So, okay. So before we take the questions, let me come back to the screen. Okay, so first of all, it was very nice presentation. It was very well explained, and I think even for the non-science people who are not from science, they must have understood the entire process of crystallization, why structure is important, why we solve the structure, and how you solve the structure of the protein, which is so important that you were able to publish it in one of the best journal in our field. So will you be able to show some of these structure on PyMol, or can we do some
Sure.
demonstration?
We can do that, yes.
Okay, so before we take the question, we still have like 10-15 minutes left. So before we end the today's session, let's have a live demonstration that how we can look at the structure, how exactly these protein look in the software, how we can play around. So if you want to practice this, you can always download a software called PyMol, PYMOL. It's a free software. You can install it on your computer and watch. Harshul is going to show you. You can just do it by yourself. It's fun and it's a lot of interesting stuff you will see. So let's see how we do it. Okay, so we are on the screen now. Harshul, if you
Yeah,
want to explain. Yeah.
yeah. So this is how the structure looks like in the software itself. You can see all these little carbons, hydrogens, oxygens that are making the amino acids. And the shape is, you can identify the protein with the shape already. This is the protein we just talked about, Rome complex. You can display the sequence here. So there are two windows in this, usually. This is the terminal. You can write codes here if you want, or you can play with the, how do you say, with the buttons that are given here. So when you go to the PDB, Protein Data Bank, you download a structure. You can go open here, and then you can open your structure. Usually the extension of the file is .pdb. Then when you have that, you can open it. This background doesn't, I don't like the background so much. So let's change the background first of all. We
Yep. So
can
this
make,
software gives you a lot of option to play around the
yes.
color, background, the way you
So,
want to see it. A lot of
and now we would like to see it as a cartoon. So what I did was I went to H, that is hide, and I clicked on hide everything. And then what I want to do is you can go to action, preset, and pretty. So all the different, how do you say, all the different domains of
moment.
the protein are shown in different colors now. Okay, so this was the B part. Okay, you can move it like this. You can zoom in, zoom out. This was the B part here, and you can see that this is the one B part. This is the other B part. You can go around in the sequence here. You can click on any amino acid; it will be selected. Then you can go to the. So it also shows the tag that I was using. That was there. Then you can click on the. What do you say? Click on the substrates that are bound. So this was one of the substrates that is bound. You can see, you cannot zoom in without the mouse. So this was one of the substrates. Can you see this one that I'm pointing with my screen or no? My
Yeah,
pointer is not.
I think it's too small to see the pointer.
Okay.
Maybe yeah, we can see it. Yeah.
Okay. So here is, this is the molecule that's binding here. This is the pocket that binds to that molecule. Even though the molecules are very similar to each other. So this, if you look at this one now, this one, right? I'm turning it. If you look at this molecule, this one and this one are not so different to each other, these blue ones, right? But still, this one will not bind here. It has to be so close. The interaction has to be so tight. And this is, since this was a very big protein, we only got it until 3 angstrom. But when you really want to see all the interactions that are happening, the closer to 1 you are, because one angstrom is the resolution at which you can see the hydrogen bonds, the better it is. But here you cannot see all the different ionic interactions, the different hydrogen bonds, which are coordinating the different substrates because of the resolution. But usually when you get at a higher resolution, you can see that. So then, yeah, you can change the color, you can rotate it, you can zoom in, zoom in. For zooming in, I would need my mouse that I don't have with me right now, because you have like 3 buttons, and with the mouse you can cannot zoom in. If you
So,
don't like one of
so
the
how
chains, you
this is the structure you solved, right?
yeah,
So for the
together
viewers,
with
it's
the
yeah, so it's so nice to see the protein which existed for, like, thousands of years. The protein whose structure was not solved for last 100 years. She was able to completely find the structure of this protein, and it looks like a loop, and a lot of but there is a very fine detail. If you want to zoom in and look at the molecular level, you can see each hydrogen, each carbon,
yeah.
each nitrogen. You can see it's a complete molecular structure. I mean, we are trying to present it in a form of very simplistic way, and it looks like, oh, it looks like that, but it takes a lot of hard work to get into there. And this is the first time she was able to reveal the structure of this very important protein, and that helped us to understand the function of this protein, how it controls the pathway she was talking about. Harshul, is it possible to take open any simple protein? Because kids, they
Sure.
might get intimidated with this really big protein. If you want to open a very simple protein, so that we can quickly
Uh,
show how the helix. And if you want, I can give you one PDB file or something to,
yeah, you can. I'm thinking which one.
or a very
Maybe
simple
this
one.
one. Okay. Yeah, this is also a small one if you want.
So, yeah, so we can hide this one, and then
Yeah.
so that we
So
can at least.
then sorry, go ahead. What were you saying?
Yeah, go ahead, go ahead. Now you can show, because I was thinking if we have a simpler protein to show, we can
Yeah.
easily show that these are the helixes and these are the beta sheets. And if you put it in a surface model, you can show that how the pockets you're talking about looks like, if you can find any
Yeah,
groove.
sure, we can do that. So this is, I think, the two domain protein from the same pathway, which is present in plants.
Okay,
And
good.
then I think I'll go on show. And if you wanted to show the surface, this is how the surface view would look like. Okay, we
Okay,
didn't
yeah,
hide everything. So we
yeah.
hide everything and then we can see it a bit better. That's how the surface view would look like.
So
And we
if
have,
you rotate a little bit, for the viewers, you can see there are many small grooves, but they might not be big enough to make a pocket in which a drug can sit and bind, interact,
there
and
is,
affect the activity of protein.
there
But in
is, I
some
think I should
are
change the
really big
transparency.
pockets. Yeah.
If I change the transparency, I might be able to see something because there is, I know that things are bound here. Can you change it? No. How do you change it? The transparency that I don't remember right now. Uh-huh. So, but if I show you not the surface,
Yeah, you
but
can
the
cut.
cartoon,
Yeah.
then we can see where is the, okay. Why is this tricky mate not shown? This is not the best structure. I don't see everything which is written here.
Would you like to put it in rainbow so that we can show which is end-valency terminal?
Yeah, sure. So,
Yeah.
now you can see. Okay. So this is the N terminal from here. That's the beginning of the protein, the blue part, and then it goes all the way here, and then it ends like at this green part. Okay, so it's coming all the way here, and it's ending at this green part. That's one chain, one part of the protein. The second part is this one.
Okay.
And the ligands, the substrates are bound. You can see that they are also part. So what you see here is exactly what we could fit in the structure. So this is a model based on the pattern that we got from the diffraction of the crystal.
Mm-hmm.
So you can be sure that it is there, not just like somebody putting it there. There are very rigorous measures when you deposit your data in this
Mm-hmm.
protein data bank. You have to answer like all the questions about your statistics. You have to give uh, how the data was collected, and so they can really see whether you're trying to fit something which is not there and you are just imagining it. So that is usually not the case.
True.
So it's always like when you see something, it's more or less definitely bound there.
Okay. So I think it's is there something more that you want to demonstrate on the?
I
I think.
wanted to tell something. There is a website called Foldit,
Mhm.
Foldit. You
So
can go there and you can install the software. It is for anybody who wants to learn about structural biology. You can go there, and when you are done with the basic, it's like a game. They make you solve the structure themselves. They will tell you, okay, how will you make this protein more stable? And then they give you tips and tricks, and until you can understand how the structure folds on itself, then they ask you to solve the problems for scientists. So once you understand the basic concepts, you can go up to this. It's really fun. Anybody can do it. You don't need anything special. You just have to make an account and then download the software. It's called Foldit. So if somebody is interested in trying this stuff out, you can also look there.
yep. So it's a lot of fun to look at the protein structure. You can try to open a protein and DNA complex, protein complex, protein lipid complex, all the biomolecules. If say for example you want to know about any protein and you think, oh, I want to know how the protein looks like, how it must be working, you can always try to find whether the structure of that protein is solved or not in the PDB, as Harshul explained. You go to the PDB. I can provide all the links in the description and search for that protein. If you find that protein there, it means the structure is solved. You will get the code for it, and then you can open that in the PyMol. Probably we can try to do one workshop also for this PyMol session, and we can walk you through the whole process of looking at the structure of the protein and doing some basic stuff. But you can always do it either on Foldit, or you can try by yourself on the PyMol. So I think that's it for the today's talk. We might have some questions, but we are already 1 hour. We completed 1 hour, so probably we will not be able to take questions right now. But what we're gonna do is, if you have any question, if you are watching it live, if you have any question, you can always post it here. This video will be uploaded on my YouTube channel, so you can, if you are watching it after the live session, you can always ask your question in the comment section, and me and Harshul will try to come back and answer those questions whenever we get time. But we promise you that we'll answer your questions. We'll not let you go without the answer. And with that note, I want to thank Harshul for giving your time. It was really nice to see you after 10 years or something,
I think,
after many years.
yeah, a
Yep.
lot of you.
2011, 12,
Yeah,
that's where
yeah.
we were doing masters. And so it was pleasure meeting you after ages. And I wish you all the best for your research. You are doing outstanding work, and I'm very sure that you will, keep making, you'll make us even more proud in future and publish much more interesting research, and make a big contribution to the science and research as such.
Thank you. And I also want to thank you. You're taking this time and doing this every week with your work. It must be like really hard. So you are really dedicated and taking this time every day, taking this time always, and doing this to teach our students. I tried to do this once, but it was not possible for me. It's really hard work, and you are very
Yeah, very
dedicated to it.
tough. So for that, I think the credit goes to my wife, because she's helping us, helping me to make this happen, because it's a lot of work to do, bring researcher on board, and arrange these meetings, and we are doing it weekly. So it's not like we are doing once in a month. So we are doing it weekly. So it's a lot of, yes, you are right. It's a lot of work, but I think. Everything depends on the student if they want to take advantage of it. It's all free. It's every Saturday. Just you have to come on the channel 8 p.m. every Saturday and just enjoy the show, because it's not about biology, it's not about physics, it's about science as a whole. And it's not necessary to just learn about one thing. You can just get glimpse of what's happening around, like how people are finding drug, how people are finding vaccines, how cancer is, people are dealing with cancer, diabetes, astrophysics, mathematics, how, all those chemical, like, in the chemistry, how people are coming up with new molecules. In material science, they're coming up with new battery, there are a lot of research happening everywhere. So having the holistic picture of what's happening around the world is really important, so that you can decide if you want to become researcher, which field you want to become in researcher in, or you can make a contribution in future once you become a researcher in future. So I think all you need to do is just hit the subscribe button. That's what I always call. Press the bell icon, because I will not be able to individually inform you that what is the next show. So just hit the bell icon so that you get notified and just watch our show. And with that note, I also want to announce that the next talk we're going to have is very interesting, because we have one scientist from NASA, and he's going to talk about the astrophysics. I'm very excited about it. I don't know about you guys. Do join us for the next talk, which will be next Saturday. Thank you, Harshul, for joining us, and I wish you all the best for your future.
Thank you. Same to you.
Take care then. Bye bye.
Bye bye.
Yeah, take care. Bye. Bye, guys.
Bye.